Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
So I think we can start now the second part of the afternoon. We have a joint talk now by Jonathan Carlson and David Eckerman from Microsoft Research. And the title of the talk is Graphical Models for HIV Vaccine Design and Genome-Wide Association Studies.
Thank you. Is this on? So rather than doing a joint talk, I think we'll do it in sequence so that you can actually hear what we're saying. I'm going to talk about the HIV vaccine design part of this, and then if I leave David enough time, he'll talk about genome-wide association studies. So I'll title my part of the talk, Learning Phylogenetic Dependency Networks for HIV Adaptation. I think we'll go into these dependency networks in a bit. I think the intuition kind of is clear from the word. But I'm really going to dwell a lot on the HIV adaptation as an application domain because I think it's very interesting and hopefully will prove to be very practical. But it sort of motivates sort of what's underlying the dependency networks that we're trying to build. But I think you'll appreciate these dependency networks are quite general. And I guess coming after Joe's conclusion that phylogeny is specifically really only between species, certainly we're going to be talking about HIV, which is not the case, but it turns out we can use the same models in this case for viruses because they have low recombination rates. So the nice thing about using HIV as a motivator is it doesn't need much motivation. We all know that it's been quite the pandemic. And certainly we don't have any solutions to this disease, not for lack of effort or money or the amount of time that we've put into this, but really because of the underlying nature of the virus and that we still just really don't understand it very well. Certainly vaccines are the ultimate goal as far as third world is concerned. And to date we have roughly 175 vaccine trials that have been run, and none of them have been successful. In fact, if you've been following the news at all, just this last September, Merck did an emergency cancellation of a major HIV vaccine trial that was actually a sort of unvaccine in the sense that people who were vaccinated appeared to have a higher chance of getting the virus and getting a worse version of the virus. So suffice it to say, we don't really understand the underlying mechanisms of what's going on here. There's a lot that needs to be done. And it really comes down to HIV's amazing capacity to adapt. So I'm going to spend maybe more time than usual on the introduction so that we really understand what's going on with the biology, because as I said, I think it really motivates the underlying models that we're going to use, and then really go into these dependency networks and what they mean. So HIV adaptation is, in a nutshell, it's what leads to all of our headaches with this disease. It's the underlying causal mechanism of drug resistance, vaccine failure, and ultimately AIDS. So lots of ways we can think about adaptation in general. For viruses, it tends to be a little bit simpler in the sense that the adaptations really are specific amino acid substitutions or sets of substitutions that confer resistance to a drug, for example, or confer resistance to your specific immune system. And one thing to keep in mind is that a person is infected with a population of HIV. Typically, our understanding right now is that you're infected with a pretty unique HIV virus, and then through time, because of the rapid evolution, rapid mutation rate, you end up with a population of virus, and it goes through the normal ideas of amino acids coming to fixation based off the selection pressure that's unique to the individual patient. So what drives selection pressure? Well, of course, that's just the or what drives adaptation, of course, is just a combination of variation and selection pressure. And HIV is relatively unique in that the variation is really quite phenomenally high. So if you take a cohort of several hundred patients, it's not uncommon to look at pairwise sequence similarity measures of only 90% amino acid similarity. This compares to estimates of human to chimp of 95%. So this is a very variable virus. It can tolerate a lot of change. And then, of course, the selection pressure is what drives adaptation. So let me start with just a basic overview of how HIV works. HIV in this cartoon is this round blob here. And it infects the host cells, the human host cells, and then goes through this process of reverse transcription where its RNA, which is what its genome is reverse transcribed, inserted into our genome, and then it takes over the human machinery, and the RNA, or the vaccine, or the HIV virus is actually transcribed, the proteins come out, and eventually the cell makes millions of copies of this new vaccine. So essentially the virus is hijacking the cellular machinery to create new copies of itself. And really the key point here is that this reverse transcription is highly error prone, and this leads to this rapid mutation rate that makes it so that it's been estimated that on the order of millions of distinct copies of the virus are generated each day in each patient. So it has a tremendous capacity to adapt. I'll skip over that. So just like the HIV adapts to our immune systems are adaptive. And there's really two arms of this adaptive immune response. The first is the humoral arm, which are the antibodies that we usually think about when we think of the adaptive immune response. And the goal of the humoral arm of the immune system is to clear foreign particles that are floating around the blood or the lymph system. So they target these free floating virus particles. The problem with HIV is that the surface is almost entirely made up of human proteins and phospholipids, and there's only a single HIV protein on the surface, and HIV is clever, and it decided that that protein would be highly variable and has only a few amino acids. So it's a constantly shifting target. It's very difficult for the immune system or for a vaccine to target that. Now, every vaccine to date that has been successful in other viruses have targeted the humoral arm of the immune system, and the vast majority of HIV vaccines have done the same. And part of the reason why they've failed is because of this property, that there's so few targets to go after, and those targets are highly variable. Now, the second arm of the immune system is the cellular arm. The goal of this arm of the immune system is to recognize human cells that have been infected and destroy them before they can generate new virus particles. And the way it does this is through a complex signaling mechanism, which I'll describe in a second, whereby the cell broadcasts to the immune system what foreign particles are circulating inside of the cytoplasm. So it basically says, hey, here's a foreign particle that's supposed to be here, and if not, then the cell is destroyed. And this is really, the hope is that this is the new frontier for vaccine design. This was actually the Merck vaccine was targeting. It was the first vaccine to target this, and it failed miserably, which sort of highlights our lack of understanding of this system. So how does that system work? Really the key player here is this protein called an HLA molecule. It's a human protein. And what it does is it recognizes short epitopes. These are short protein sequences belonging to the virus. Recognizes these epitopes, transports them to the cell's surface to broadcast their presence. Cellular immune response, these T cells learn to recognize the epitope HLA complex. And when they recognize it, they destroy the cell to prevent it from generating new virus particles. Now, the important part here is how the HLAs and the epitopes interact. So these epitopes are short strings in the particular class of HLA molecules. They're like 8 to 11 amino acids long. And each HLA type can recognize a specific set of amino acids. Now, it turns out that these HLAs are highly polymorphic. Right now, we've identified some 1,000 or so different HLA variants in the human population. And each one, like I said, can recognize a specific subset of these epitopes. And it's still a very open problem as far as which HLAs recognize which epitopes. The other important thing is that there's 3 loci. So there's HLA A, B, and C. And so we have 6 HLA types, 3 from mom, 3 from dad. And this means that each of us will fight any virus very differently. Now, there's 2 key ways that HIV can adapt to this selection pressure that comes from the cellular immune response. The first is to select for a mutation within or nearby the epitope that prevents the HLA from ever binding to the epitope. If the HLA can't bind the epitope, it can't alert the immune system to its presence. And the second is a mutation that prevents the T cell from actually recognizing the epitope. In the end, it has the same effect. This mutation makes it so that the immune system can't see this particular epitope anymore. So just to summarize the biology here, the cellular immune response learns to recognize the HIV peptides presented by the HLA proteins, but HIV can often escape via specific mutations. And really the big point here is that the HLAs mediate a patient-specific selection pressure on HIV. And one of the open questions, really up until a few years ago, was how big of a source of selection pressure is this? And the reason why this is important is it really gets to the question of, is the cellular immune response actually active against HIV? One possibility as to why the Merck trial failed is that this cellular immune response isn't actually doing much to begin with, and so there's just nothing going on there.
Why would it have made it more likely?
Well, so this suggests that something's happening, but part of it, too, I should caveat that it's not entirely clear what's going on. There could be confounding factors there. So let me give you a specific example that's been well studied in the laboratory. So HLA B57 is one of these HLA molecules, is known to recognize this 10 amino acid sequence, and is known not to recognize this 10 amino acid sequence. So there's just a single point mutation, a threonine to asparagine, and that completely prevents HLA B57 from recognizing that epitope. It turns out that that mutation also has a huge impact on viral fitness. So in the absence of cellular pressure, if you just have a virus sitting there in a test tube, if you give it this mutation, it doesn't replicate very well anymore. But that fitness cost can be compensated by 4 or 5 mutations that are 50 amino acids away. So the big picture of what we're seeing here is that when a person with B57 gets infected with the virus, B57 recognizes this epitope, the cellular immune system starts to destroy the cells that are infected, so the viral fitness is very low. The virus then selects for this T to N mutation, which prevents the cellular immune response, and so the viral fitness comes back up a little bit, but not back up to wild type because of this structural fitness cost. And then these compensatory mutations partially recover the fitness cost from this mutation. Does that make sense? So our goal then in the project is to map specific HLA alleles to escape mutations and their compensatory mutations. We want to understand what the constraints are in viral evolution and how we can train the immune system to specifically target, effectively target epitopes that cannot escape or whose escape is very costly, and or to block these series of compensatory mutations. So what are these dependency networks? Well, a dependency network is a graphical model that you can sort of think of as what happens when Bayesian networks and Markov blankets run into each other. The key point of the dependency network is that we're primarily interested in local conditional probabilities. We're not as interested in the joint probability, so this allows us to very efficiently learn these networks. And the practical aspect is that we want to say, so here's a codon here at position 17, and we know the amino acid, and we want to know what variables predict the amino acid that a patient will have at position 17. And so in this case, we have HLA A03. This is one of the alleles, predicts what what amino acid you'll have. And another codon, codon 1, also predicts the amino acid you have. So you can imagine, given one of these dependency networks, it's very easy to come to generate hypotheses about chains of compensatory mutations. And these are things that we can immediately go and test in the lab, so whether these are causal mechanisms or simply correlations. I should say this represents something that we see in sort of gene studies a lot when we want to know gene-to-gene interactions. And really the problem is, how do we account for the phylogeny? And I'll get into that in a moment. So building dependency networks, as I said, is all about local conditional probability distributions. And certainly the simplest way to think about this is pairwise associations. And as far as compensatory mutations are concerned, this is something that people have been thinking about for 30 years now. And so we'll just start there and talk about the previous work and what people have been doing in this area. Now the naive approach is to assume that everything's independent. And so here we have 8 patients. We have their sequence alignment. We're going to consider one particular protein in HIV. We're going to ask which columns of these alignment are correlated with each other. So here's this epitope that I pointed out earlier. And here's a hypothetical example of an amino acid that's exactly correlated in these 2 positions. So every person who has asparagine here has an I over here. Everyone who has T has a V over here. So we can throw up together a nice contingency table and see that that's significant. But it shouldn't take much convincing of this audience that we're missing a major confounding factor, and that's sort of the underlying evolutionary history of this virus. And here's an example that this problem has been known about for a long time. I think Joe most eloquently described it back in 1985, but let me just go over this again really quickly. So we're looking at correlations between T and the amino acid that's observed, in this case at codon 256. We have 2 different cases. In each case, everyone who has T has V. Everyone who has N does not have V. The main difference here is what's going on in the underlying phylogeny. So this is a phylogeny that we will build up from this particular gene, the GAG gene. So we call it a gene tree. But what this tree tells us is that everyone who has T essentially inherited it from somebody from the common ancestor. So likely this person had T as well, and same over here. So in effect, we don't have 8 independent cases. We really only have 2 independent cases, and we're really going to overestimate the significance of this association. And of course, the opposite is also true. If the correlations are opposed to the tree, then it's actually stronger evidence that there's some sort of causal relationship between these 2 variables than if we ignored the tree altogether. So in the case of compensatory mutations, I said there's a rich history of people investigating how we can take into account the phylogeny dating back to 1983. And it's still an ongoing area of research, but I think maybe the best model as far as intuitively what's going on was described by Allelic et al. In 1999. And the idea here is that we're going to use two graphical models to describe evolution. We're going to have a null model, which is just that the two amino acids are evolving independently. And our alternative model is a simple extension that says now they're evolving jointly. Now the independent model is we use a binary variable, and so it's just evolving down the tree. And so to go to joint evolution, that's simply a matter of taking a binary variable and changing it to a 4-state variable. We can describe that in the reversible case with just 5 parameters, and that works out quite nicely. So that's that for compensatory mutations. We'll come back to that in a little bit. But what about these HLA escape mutations? Now, one of the key assumptions of the Allelic model is that we're looking at compensatory mutations. So it's reasonable to assume that both of our variables are evolving down the same tree. In the case of HLA, that's a property of the host, which may have nothing at all to do with the phylogeny of the virus. The Allelic model, in theory, can model this by sending one of the rate parameters to infinity, but in practice, this tends not to work out so well. It's prone to local optima, and it's just slow because of the matrix math. And ultimately, we want to scale beyond just 2 variables. We want to build up a full network, and just pairwise comparisons turn out not to be very good. And this Allelic model does not scale well. The number of parameters blows up quite quickly. So the first solution proposed in a very influential 2002 science paper was just to ignore the phylogeny altogether. Say, well, the HLA is more or less independent of the phylogeny, so let's just use a chi-square analysis, which on average should give you the right statistics, but the variance of those statistics blows up, so it becomes very hard to actually interpret your results. And they found 80 significant associations, but in the last 7 years or so, there's turned out to be very little experimental support for any of those associations. So it appears to be that most of those were spurious. So we went back to the drawing board, if you will, and David and some people at Los Alamos came up with an alternative model, which is just a simple extension to the null model. Again, we're going to have null models that the amino acid is evolving neutrally. And the alternative model now, rather than joint evolution, is simply that the HLA that we observe in the current patient adds a level of selection pressure. Let me show that really quickly. Here's our null model, which is the likelihood model that Joe just described in his talk before. So again, we have 2 parameters, the stationary distribution and the rate of mutation, and then we're given the tree, which has branch lengths. And again, the model here is that the amino acid is evolving neutrally down the tree. And we're just going to use a binary model to gain power and for ease of use here. Now, the alternative model that we want is that the HLA is going to influence this last step in the evolutionary process. We don't know the HLAs in these hypothetical ancestors, so we're going to average out over those possibilities, and we're going to focus on the HLA in the patients that we actually know about. We're going to do this through a simple extension of the model.
Is this tree a tree of patients or a tree of within patients?
Sorry, so each individual here is, comes from a patient, and so we sequence the virus, the consensus sequence of the virus in each of the patients. Yes, and then we'll look one codon at a time. Yeah. And it's really the predominant sequence in that patient is the hope. Right, so our alternative model now is that the amino acid is evolving neutrally down the tree, and these orange nodes now are the amino acid that we would see in the patient if the HLA had no selection pressure at all. But now we're going to allow the presence or absence of the HLA to influence one more stochastic hop here. We're going to parameterize that by the strength of selection pressure, sigma here. So now we can have either a positive or negative selection. If you have the HLA, maybe it pushes you away from wild type to an escape mutation, for example. Does that make sense? So I think at this point it's worthwhile just comparing these two models. Again, the main comparison is that the joint evolution is assuming coevolution all the way down the tree, and this is undirected joint, so if either one changes, the other one changes. So we're looking at joint probability distribution of the two variables together. In the conditional model, all we care about is the predictability, how the tree and the HLA predicts the amino acid. So we're only interested in the distribution of the amino acid, not the joint distribution of HLA and amino acid. And this allows us to ignore the distribution of the HLA. So the HLA could follow the tree, it could be independent. It I suppose somebody could come up with a distribution that was exactly opposite of the tree in some way. And this also allows us to reduce the number of parameters to 3. And most importantly, as we'll see in a second, it turns out that this model is easy to extend beyond just 2 variables. Now you can ask the question, how do the models approximate each other? So this is a simulation study where we generate data according to either the joint evolution model or the coevolution model, or sorry, the conditional evolution model. And these were parameters that we learned from real data, and then we just, as in a parametric bootstrap, we generate data and then try and learn it back and see how well we did. These are precision recall curves. And here you can see in green is the joint model, and as you expect, it generated the data, so it does best. And blue is a conditional model, and it actually approximates the joint model quite well. And then in red for comparison is the Fisher's exact test, and not surprisingly, there's a drop in power there. When we generate data according to the conditional model, again, as we'd expect, the conditional model does best. It was a generating model. But the coevolution model in green does not do as well, presumably because of this problem of having to send the rate parameter to infinity in order to model this and issues of local maxima. Fisher's exact test does fairly well, but again, the main problem with that is the variance in the p-value. It becomes very difficult to interpret the statistics that come out. So let me just take a sidestep here and address the issue of multiple hypothesis correction. So in the case of HLAs, we want to compare every HLA allele. Like I said, there can be up to 1,000. Most of these populations is a couple hundred. Every position within the protein, HIV protein. And then every observed amino acid at every position. So needless to say, we're testing lots of hypotheses. And so we're going to use a false discovery rate, which at the end just gives you this right here. So at a given level of significance, what proportion are expected to be spurious. This turns out to be a very useful statistic for biologists who can go in and test 10 different models, and they don't care if 2 of them are wrong, but they want to have an estimate of how much utility they're going to get out of these tests. Now, in order to measure the false discovery rate, we need to understand the null distribution. As I said, we have nested models here, so that allows us to do a log likelihood ratio test to get the p-value, but then in order to get the null distribution of those p-values, in order to get the false discovery rate, we need to generate null data. And because we don't care about the distribution of the HLAs, we only care about the distribution of the amino acids, we'll preserve the tree, and instead we'll just permute the HLAs. So we're breaking these arcs between HLA and patients here, and then that will give us a null distribution. So we went back using this FDR approach, this phylogenetic correction approach, to this original study, and reanalyzed the data, and we found that rather than 80 significant associations, we found only 8 significant associations. But of those 8 associations, we were able to verify them experimentally. We used a false discovery rate of 20% in that case. Now, 8 associations from 80 is a pretty significant drop, and there was much noise created right after this was published, saying that, well, maybe the cellular immune response isn't doing anything after all. But of course, that begs the question of what's our false negative rate? How many are we actually missing? So that's pretty easy to study as well using synthetic data. Same way we generated those power curves. And we find that with 150 patients, our false negative rate is 95%. So basically don't read anything into those conclusions. What we need is more data. And so we started working with folks up at University of British Columbia, who at the time had the largest data set in the world of treatment-naive individuals. And I should have pointed this out earlier. We're interested in the genetic effects of patients, so we're specifically looking for patients who have not been on therapy. So they had up to 700 patients, and now we're seeing many more associations and really confirming the notion that the cellular immune response is quite active. Again, we can do the power studies and estimate that we're still missing about 50% of the associations. So in summary, the conditional model allows us to approximate the joint, even on joint data. And as we'll see in a second, it actually scales well. So we're going to focus on that model from here on out. What's the key limitations? Well, as I've been alluding to, the pairwise comparisons is really the problem here. So there's 2 major sources of problems with the pairwise comparisons in this domain. The first is linkage disequilibrium. So that I said there's 3 loci, HLA loci, A, B, and C, that are right next to each other on the chromosome, which means that you're inheriting B, A, or C from mom or dad is not independent of each other. If you get B57, then that actually predicts what C allele you'll get. So when we go down now and compare every HLA to every codon, if this is the real model where B57 is influencing codon 242 and is influencing CO6, then we'll also learn an arc between these 2, which is spurious. Of course, the same is true with compensatory mutations, and you can get different effects too. So in compensatory mutations, you can get chains of effects. So this was a chain that was experimentally defined where B57 leads to escape at 242, which leads to compensation in a number of other mutations. And when we're doing pairwise comparisons, we'll actually find arcs that connect these two, which are not really there. So what we'd like to do is move beyond pairwise associations to multivariate associations. And this, I think, is where things start getting a little more interesting as far as what we get out in the end. Really, our ultimate goal is to find the minimal set of predictors for each amino acid. So given the set of sequences, given the HLA types, can we predict what the specific amino acids are that will arise in a given patient? And here's our pairwise comparison model. And again, this is neutral evolution down to the patient, and then we have a single predictor. And you can imagine that it's quite simple now just to add in more predictors. So how do we do that in practice? Well, we need a model of what selection pressure is actually doing here. So selection pressure can be either positive or negative. Whatever this predictor value is can make you more or less likely to have wild type, for example, or you could have no pressure at all. And there's a number of ways you could think of combining this, but the simplest method, and what we're going to start off with, is the additive model. And that essentially says that if you have two sources of negative selection pressure, you're more likely to have net negative, etc. And the other issue is that the selection pressure is stochastic. So just because you have a B57 doesn't mean that you'll actually mount a immune response. And of course, the stochastic process you could think of as being roughly proportional to the relative fitness cost of the virus of not escaping. So we want stochasticity and we want additivity. So that leads us to a pretty intuitive model, I think, which we call noisy add. You can think of it as generalization of the noisy OR model. So in noisy OR, each of these nodes fires with some probability, and if at least one fires, we say that the sort of the collection node fires. But now we want a notion of positive and negative signs. So we have our three different predictors here. Each one fires with some probability. Some have contribute negative selection pressure, some positive. And really what we want in the end is a conditional probability of what's the probability that the net result is positive, negative, or 0. And given that, then what's the probability of a transition down here? So that's what that says. And of course, we have a question now of how do we build up these nodes? What predictors do we include here? And so the simple solution to that is just to use forward selection. The way forward selection works is we'll start off with just a single predictor. We'll run it through, test all possible single predictors, so all amino acids, all HLAs. We'll find the one that has the strongest p-value, and then we'll add that to our model, and we'll iterate. Now, when we iterate, we have a null model, which is simply the previous iteration. The alternative model is adding one more. So if we start off with B57, now we want to say, does CO6 add anything? Then we ask the question, does B57 plus CO6 predict our amino acid better than B57 alone? If they're in linkage to equilibrium, the answer is probably no. So the first question is on synthetic data, does using multivariate associations actually help? Does it give you anything beyond the pairwise associations? People for a long time have been assuming that pairwise associations is as good as we can go. How bad of an assumption is that? Well, here we generated data according to the additive model. And I've actually plotted 3 lines here. I haven't shown you the data for this pink line, but the yellow line is our noisy add model. This pink line is another approach that we did based off decision trees, which is a little more general and actually could be applied to Pollock. But I don't have time to go into that today. And then the blue line is what happens when you just do pairwise comparisons. So you can see an absolutely enormous jump in power once you start considering the multivariate effects. And this is really because we pick up a lot of spurious associations every time we pick up a true one. And we can look at calibration as well. So when we predict the false discovery rate is the Q using this null distribution, and we compare it to what the actual false discovery rate, if you will, is. We expect to see a straight line, and again, our noisy add model is slightly conservative, and we see this consistently over a lot of different runs. Whereas, not surprisingly, if you just look at pairwise comparisons, then you get highly inflated statistics, again, because every time you find something real, you pick up something false as well. So what do these dependency networks look like in real life? So this is HIV protein GAG, which is people think will be very relevant for vaccine design. And I've drawn the protein sequence in counterclockwise order here. So this is the start of the protein, the end of the protein. And all the external factors I've drawn on the outside. So these are the HLAs that influence specific amino acids. And then compensatory mutations are drawn across the circle. Unfortunately, there's not a lot of known associations. There's maybe 3 or 4 in GAG where we know the actual HLA to amino acids and the compensatory mutations as well. I didn't put it in this figure, but you can drill down, and all of the known ones that we've talked to researchers about, we actually reconstruct the actual process. So we find B57 influences this codon, which influences this codon over here, which influences these codons over here. And that was sort of the model that was just published this last fall for escape of this position. So let me summarize then about the multivariate analysis. And this isn't surprising. People have been talking about this for a long time, but it's just been difficult to actually do this where we account for both multiple inputs and the phylogeny at the same time. You typically do one or the other. And not surprising, accounting for them both increases power dramatically. I think I'll skip over this in the interest of time, but suffice to say we're working with a lot of biologists as far as actually taking these predictions and testing them and what can we get out of them. But with that, let me take questions and then pass it off to David so he has time here. Yeah.
So in your simulation study, when you simulated joint versus from joint model of the evolution of the traits versus your conditional model, the differences between your method and chi-squared, naive chi-squared test were not too significant. They were close.
Right. So
surprisingly close to each other. But in your data, when you apply it to the science paper, you have a huge discrepancy between those two methods.
So part of that is that I was plotting a precision recall curve as far as the actual false discovery rates, false positive and true positive rates. And the problem is that the chi-square, the statistics are not calibrated, so it's very difficult to know what the actual values are. That's really what it comes down to.
Thank you. Does anyone not know what a genome-wide association study is? I heard a yes. I'll be very brief. What do I have, 5 minutes? 15. Okay, I will not take 15 minutes. I think we all appreciate that both your genetics and environment influence various things that might happen to you. Will you get a disease? When will you get the disease? Will a particular drug treatment work? But we haven't had data to look for the correlations between genetics and these things until now. Now you can take someone, for $500, you can sample 500,000 different nucleotides in an individual, and this data is becoming widely available very quickly. And so with this data, then we can start looking for correlations between a person's genetics and various things like this, thus enabling personalized medicine. And this is happening now. In February, the first genetic test was approved by the FDA. It's called MammaPrint. Based on your genes, this test can tell you with a fairly high degree of certainty whether a particular form of breast cancer will recur in you or not. And that's very important to know because there's a drug that treats for this particular type of breast cancer, but it has very bad toxic effects. So it would be very useful to know from a test of your genetics whether that disease will naturally recur in you in order to decide whether you should take this rather toxic drug. So this is the tip of the iceberg. We're going to see lots more of this happening very rapidly now, I think. Now, how do you look for correlations between genetics and phenotypes? One way to do it is just scan the entire genome of a human. These are called genome-wide association studies. So you take all of a person's DNA and you sample these 500,000 nucleotides, and you usually aim for nucleotides that tend to vary a lot among individuals. These are called single nucleotide polymorphisms, or just SNPs. And now you take 1,000 people who have some disease and you measure their SNPs. Take another 1,000 people who don't have the disease, measure their SNPs, and then look for correlations between the SNPs and this particular phenotype. And then you can take whatever phenotype you want. Do they respond to a drug? Will a disease recur? Whatever you like. So the main problem with these studies is that, as you might guess, the data is very noisy. So you want a method or you want to use statistical methods that have as much power as possible. And I want to go back to a version of Jonathan's slide before, and the main point of this slide, or one of the main points of this slide was, if you take population structure into account, if you take the sequence structure into account, sometimes you can get more signal out of the data than if you don't take that population structure into account, namely in this case here that Jonathan showed you. So we thought, well, we've developed this method for HIV evolution. Let's see how it works with genome-wide association studies. It seems like roughly the same problem. And here is Jonathan's model again. I'm sorry, this is in black and white. It's not in color. But we're trying to, this is just the univariate version, the pairwise version, trying to predict an amino acid from an HLA. Well, let's get rid of the amino acid and replace that with phenotype. Let's get rid of HLA and replace it with SNP. You can certainly do multivariate versions of this and look for multiple SNPs or have other demographic information about the patient here. But now you've got a fairly decent model with perhaps one exception. Remember before these hidden variables here, they essentially represent everything you know about the patient's sequence with respect to the other sequences in terms of predicting the target variable. Here, and in the HIV case, we represent the relationship among these hidden variables with the phylogeny. That makes sense for HIV, which is monozygotic, if that's a word. And it's not a very good assumption for humans who are dizygotic. So we have to substitute some other graphical model here to represent these relationships. And this summer, working with Hyun-Min Kang at UCLA and also Jonathan and Carl Cady, we came up with what we think is pretty much the simplest possible model for the relationships between these hidden variables. We just assume there's a single hidden variable per person, and again that variable is meant to represent everything we know about that patient sequence in relationship to the other sequences for predicting the phenotype. So we have one hidden variable per patient, and then we assume that these variables are related by a Gaussian distribution. And we need to put some covariance matrix on this Gaussian distribution, and we just construct this covariance matrix by looking at the SNP data for each of the patients and building a similarity matrix. For those of you that know, it's an IBS, identical bisimilarity similarity matrix that we use here. So now we've got this Gaussian distribution among the hidden variables, and we use that, and again this Gaussian distribution is constructed from the SNP data of the individuals, capturing this population structure among the individuals. And now we use those hidden variables in combination with the SNP to predict phenotype. Those of you that are familiar with graphical models will know that this is difficult to do because this is a continuous variable and this is a discrete variable. So we need some link function between them. We chose the logistic. And then to learn the parameters, you've got to do inference in this network. And you basically can't do that in a tractable fashion. So we introduce a variational approximation where instead of using a link function here, the logistic link function, we approximate it as a Gaussian, but we fit the Gaussian so that it fits the logistic function as best as possible. And then you end up with a generalized EM algorithm that can learn these parameters quite easily. When you do that, here's what we get. This is an ROC curve. Sorry, Jonathan used sensitivity specificity. I'm using true positive rate versus false positive rate. So in Jonathan's curves, you want to be over here. In my curves, you want to be over here in this region. But anyway, if you look at the Fisher exact test, which doesn't take into account structure at all, you get a curve like this. By the way, this is based on synthetic data that's generated from the model that I just showed you. So if you don't take into account population structure, you get a curve like this. If you use a popular method called Eigenstrat that to some extent takes into account population structure, you get a little bit better. And then when you use our model, you get very good performance. This is not a big surprise. We generated the data from this model, so we expect to be better. But now this is encouraging, and what we're in the process now of doing is taking this model and applying it against known real data sets, and then we will go off and try to validate some of these presumably additional associations that we find. I think that's about it. I'd like to thank a lot of people here. Most of these people are involved with the work that Jonathan mentioned. Sabrina Broom provided that large data set that Jonathan was talking about. Several other folks at Mass General have contributed as well. Folks at Microsoft Research, we're working with, in addition to the people I mentioned already, Chris Meek and Ido Wexler on these genome-wide association studies. And also people elsewhere, especially Richard Harrigan, who also contributed one of these large data sets that Jonathan mentioned. Okay, thanks. Any other questions? Yes.
So in the last what was the model that was, because the eigenspread people sort of have this very continuous admixture model, which gives you the eigenspread model.
Yeah.
So what was exactly the model?
And what they use are mixtures of, or a PCA model, like a factor analysis model, essentially. And we use a full covariance matrix generated from IBS. So we're not trying to factor that full covariance matrix into a few spherical components. We're just taking it as it is. I don't think that's why we do better, though. I think we do better because Eigenstrat, let's see. I haven't verified this. We're in the process of verifying it. But in this model, when you have data here, it can influence the inference or the signal from this data can propagate through the whole network through these hidden variables. Eigenstrat doesn't have that property. And so in a sense, all the data here is informing all the rest of the data or all the rest of the parameters that you're learning throughout the network. Eigenstrat doesn't do that. I think that's why this works better.
And just one other comment is, what about the power of the test? Because I think if you actually do a GWA test, we'll take any spurious correlation that you can.
Oh, yeah. No. Yeah, if you do, I didn't show it, but if you do QQ plots on this, they look beautiful. They're just as flat as Eigenstrat. And so this difference here in the RLC curve is coming from power. Yes?
Scanning along the genome, are you making use of any coalescence relationships?
No. This simple model that we came up with was the simplest thing. We wanted something really easy that we could do inference with.
So it's sort of an empirical model instead of
It's absolutely, and I think a coalescent model, if we can figure out a way to do inference in it in a practical way, I think that would be a very nice thing to try here.
What's the size of the matrix K?
Well, it's the number of individuals you have. So this is, typically genome-wide association studies have somewhere between 1,000 and 10,000 individuals, so it's 1,000 by 1,000 or 10,000 by 10,000. Okay.
Thanks. We have another talk before the next coffee break, and the talk is by Yoon Song from Berlin. And the title of the talk is on the genealogy of asexual diploid.
Okay, sorry for the trouble. So this is joint work with Chuck Langley, a population geneticist at UC Davis. So today I'm going to tell you a bit about a problem which has been, we think, largely ignored by the phylogenetics community. And this problem is actually very tightly linked to a classic issue in population genetics. So I'm going to begin with a rather long introduction. Perhaps you will find that most interesting of this talk. So here are some more provocative titles on this issue. The issue that I will try to address in this talk is the apparent prevalence of sexuality in nature. So there are about 2 million named species to date. And of these 2 million species, only 2,000 appear to be completely asexual. So on the face of it, this is quite surprising, since reproduction in asexual organisms are usually easier than that in the sexual counterparts that undergo meiosis and mating. So what do I mean by that? So in a diploid organisms like humans, each individual has 2 copies of each chromosome. One comes from the mother and the other from the father. And each copy is a mosaic of the 2 copies in the corresponding parent. And the recombination is the process that creates this mosaic pattern, as Dan talked about this morning. In contrast, in an asexual organism, you just have a cloning, it's mitosis. So you have a mother and you have a child, which is obtained by cloning, self-cloning. So it doesn't require fertilization. So this is much more efficient. It costs less energy, and it can be done much faster. But nevertheless, this kind of mating and reproduction is much more prevalent in nature. So why sex and recombination? So several ideas have been put forward to explain this phenomena, usually invoking either epistasis or interference of selective forces at different positions in the genome. But it still remains debatable as to which precise evolutionary conditions and mechanisms maintain sex and recombination in natural populations. But I think most people would agree that sex and recombination are important for the long-term evolutionary success of an organism. That is, asexual organisms are much more likely to go extinct than the sexual counterparts. So this is a widely accepted wisdom, but there are counterexamples in nature. So I think the best known example is the diploid rotifers. So they are microscopic aquatic animals which are found throughout the world, and it has diversified into 4 families, 18 genera, and 360 species. Just to remind you, this is the biological classification scheme. So on the top, you have superclasses or supercategories, and as you go towards the bottom, you have subcategories. So you get finer and finer as you go down here. And diploid rotifers are over there. Everything below this lineage, everything below here has remained asexual. So this is quite surprising. Just to give you an example, let me just contrast that to Nematophorus. These are more commonly known as whiptail lizards. And shown here are 3 different species of whiptail lizards, Nematophorus. And exactly one of them is asexual. So would anyone like to take a guess which one is asexual? Oh, John, you're not allowed to guess. But that's right. It is the middle one. You just knew the answer? Or? Okay. That's right. It is the middle one. So this one is asexual. So it's quite surprising that everything here below this lineage has remained asexual for over 40 million years, tens of million years. And molecular genetic analysis suggests an age which is close to 100 million years. So these are some examples of diploid rotifers. There are 4 different species. So as I said, fossil evidence suggests an age which is between 35 and 40 million. But molecular genetic analysis suggests an age which is twice as large. So this one here is digesting food, and this is a well-developed egg. I don't know what they are doing, but they're doing something. They have nerve ganglia, muscles, digestive organs, and ovaries, and so on and so forth. So Maynard Smith, Sir Maynard Smith, referred to diploid rotifers as something of an evolutionary scandal. And it has been questioned in the past whether they have indeed remained asexual for all that while. So the question I want to pose is, so how do you actually estimate the age of asexual organisms? I mean, how do they estimate this number? And the point that I want to make is that there is no proper phylogenetic method for doing such analysis. And I think this is a really interesting problem, but hasn't been really explored. So I'll get to more details. But there are counterexamples to the fame of ancient asexuality in the past. So a scale insect species, once thought to be asexual, ancient asexual, turned out to have very tiny male forms that happily live being attached to the female's leg. This is not science fiction, this is real science.
It happens in real life, too.
I see.
Except that the females have tiny.
I'll let you sort that out. But anyway, the beginning of molecular genetic evidence really began to emerge in the year 2000 when Meselson and his graduate students analyzed molecular genetic data from the 4 diploid species that I showed before, those 4 species. And they provided some evidence in support of diploid lodifers, ancient and continuous asexuality. So this is their method. So you take 2 copies of a gene within an individual. So 1 copy from one chromosome, the other from the other chromosome, the same gene, and you count sequence differences, Hamming distance or edit distance. At how many positions do they differ? So on the neutrality, so if you look at those sites which do not change amino acids, then maybe they are on the neutral evolution, then the differences are expected to be overrepresented in an old asexual organism in comparison with that in the sexual organism. And the reason being the genetic drift, this random fluctuation is more efficient if you have sex and recombination. If you don't have genetic drift, then you're more likely to accumulate differences. So this is a schematic depiction. So you expect to see more differences in asexual organism than in sexual organisms. And when they applied this idea to diploid lodifers, they observed the following. So they looked at diploid lodifers and its sister class, monogonal lodifers. Let me just remind you the classification scheme. So there was a classification scheme. The diploids are here, and monogonals are just another class on the lodifers. So they looked at diploid lodifers, which are asexual, believed to be asexual, and they looked at monogonal lodifers, which are partially sexual. They seem to reproduce sexually occasionally. So this is just a schematic depiction, but this is exactly what they actually observed in real data. So those are the positions where they differ. 3 positions, but in diploid lodifers, a lot more. So that's like 0.0% to 2.4%. Here is between 30 and 73%. So another question, well, how often do these guys reproduce sexually? Right now, there is no method of estimating this parameter. So we think they reproduce occasionally sexually, but we have no idea how often. So this was a great success, but if you apply the same method, same technique to different ancient asexual, or supposedly ancient asexual organism, then this method fails. It does not produce any significantly high level of sequence divergence in the supposed reputed ancient asexual, because you don't see this kind of high number. So what's going on here? Is it that the method is incorrect, or it's not really proper, it's not sufficiently good? Or is it that these ancient asexuals, or actually not ancient asexuals, but actually they reproduce sexually? So as this example illustrates, I think a more refined method that makes better use of DNA data is needed for studying asexuality. So could it be that sexual reproduction has actually occurred in the history of diploid lodifers and of other reputed ancient asexuals? And if so, how big a role did it play in the long-term evolutionary success of those ancient asexuals? If not, when was sexuality lost, and how many independent times? and those species that are partially sexual, how can we estimate the frequency of sexual reproduction? So we need a method of studying asexuality that explicitly models the evolutionary history of asexual diploids. So here is what's going on. So this fat tree represents the evolution of individuals, and embedded in it are two trees representing the genealogy of the two genes. You have two copies of each gene. So how can we carry out some analysis here? Well, the first thing to try would be just take one sample per individual. Well, if we do that, then you may actually make a lot of mistakes. For example, you may end up with different topology, a topology which is different from the individual topology. Okay, then what happens if you take all the sequences and try to build a genealogy? Well, in that case, you are ignoring a lot of information here. The part of the genealogy here is actually correlated with the genealogy here. If 1 and 2 merge, then these 1 and 2 should also merge. And in fact, you may actually end up with different topology for the two different groups. And this actually has been applied in the Meselson paper. They actually use this kind of technology, the standard phylogenetics method, to build genealogies and to make inference. So that is not the right thing to do. You should try to think about the coupling of the two genealogies embedded within this individual genealogy. So I think by studying the coupling, we can get a lot more information out. But there's a big question here. So given a set of sequences, pairs of sequences, how do we know which one belongs to the blue group and which one to the red group? That is the main question. Once we know them, we can do some analysis. But a priori, we have no idea. So we first need to determine, given a set of pairs of sequences, we need to partition the data. How do we do that? And once we have this partition, how can we construct the genealogy of the individuals, asexual individuals? In what follows, I try to describe a very clean method in the case of infinite sites model of mutation, which Dan mentioned this morning. It's very clean and nice, but sometimes that translates to being not very useful, and I'm very fully aware of that. But so far, we have no results on this kind of model, so this is really a toy model from which we can hope to gain some intuition. And eventually, I would be very keen to develop a genealogy-based statistical method which models the evolution of asexuals. And hopefully, some of you will be interested in undertaking such a task. So on the side, I just want to mention that this kind of genealogy is exactly what we see within ourselves. I mean, our cells reproduce by mitosis, and they're asexual too. So one can actually use this kind of methodology to study the evolution of cell clones within a single individual. And I think it has some interesting applications to cancer biology, studying the genealogy of cancer cells. So there are two types of data which I will consider in this talk. The first type is this SNP data. There's two haplotypes per each individual. It's a binary string of length m. And these two haplotypes are called mates. So I have a mate pair, and I have n individuals. So we have seen this kind of data earlier today. Perhaps some of you are also aware of this kind of data. It's called genotype data. So this kind of data is actually easier to obtain in reality than this kind of data by experiment. So this is the encoding. If you have two copies of zeros, then you encode it by 0. So this is called homozygote. You have two identical copies. This is another kind of homozygote, two copies of 1. Then you encode it by 1. If you have 0, 1 or 1, 0, then you encode it by 2. That's the encoding scheme. And this kind of data are called genotype data. And this provides a solution, a phasing solution to this kind of genotype data. So is there any questions on the definition? No? Okay. So as Dan mentioned this morning, this perfect phylogeny is a very well-studied problem. And then, although it was published in 1991, it then actually had these results dating back to early 1980s. So there's a linear time algorithm. It's a linear in the data size. M is the number of columns. N is the number of rows. And there's a linear time algorithm for constructing perfect phylogeny in the haploid case, when you don't pair up the haplotypes to create a single individual. If you're gonna the information, then you have a linear time algorithm. And the kind of objects I will be talking about today are diploid perfect phylogenies, which are just natural extension of haploid case. So instead of labeling each vertex by a single haplotype, I will either label it by a pair of haplotypes or a genotype, depending on the input data. So again, as in the haploid case, we have only one mutation per site. And when there's a mutation, it mutates only a single copy of the two haplotypes. For example, here, there's a mutation at site 1, then I only mutate the bottom haplotype, not the top one. So just mutate just one of them. And when there's a mutation event, the mutant allele has never been seen before. So this is a natural extension of infinite sites case to the diploid setting. And there's one-to-one correspondence between the leaf labels and the input data. And I will say that a DPP, diploid perfect phylogeny, is minimal if every interior edge has at least one mutation. So this is a minimal DPP, since this is the only interior edge and it has a mutation. That's the minimal DPP. So there are two problems which I will consider. The first problem, given a input haplotype data denoted by H of n individuals, determine whether H admits a DPP solution. If so, find one. Second problem, if the input data are genotype data for n diploid individuals, then determine whether G can be phased to haplotype data that admits a DPP solution. And if so, find such a phasing solution and the DPP. So those are the two problems. And we found, again, linear time algorithms for both problems. So if a DPP does not exist, then it may be interpreted as evidence of sexual reproduction, recombination, or multiple mutations, recurrent mutations. So that's the motivation. So the first case for the haplotype data. So there are very well-defined properties imposed by the one mutation per site condition. It's so restrictive that you can actually uniquely determine the root sequence. So let me just tell you how that goes. So you have two haploid perfect phylogenies embedded in the diploid perfect phylogeny. And the set of mutations in the two trees are disjoint by the one mutation per site condition. And if the root haplotype of one tree and the root haplotype of the other tree differ at site K, then neither tree mutates at site K, again imposed by the one mutation per site condition. And these properties combined can actually uniquely determine the root haplotypes as follows. So if no distinct So if two distinct haplotypes, homozygous appear, for example, 1, 1 and 0, 0, then you immediately can conclude that there's no DPP solution. If there is a homozygous individual, then you assign that genotype to the root sequence. So 2 copies of zeros, then you just assign 0, 0. 2 copies of ones, you just assign 1, 1. Whenever you see 2 copies of zeros and 2 copies of 1, 1, you conclude no DPP. Simple enough. But now suppose that you have some sites which are completely heterozygous, which means everyone in the data set, in the input data, is heterozygous at the site. For example, here, everyone has either 1, 0 or 0, 1. So it's completely heterozygous. And EH denotes those set of sites at which every individual is heterozygous. So EH here in this example is 4 and 5. It's a set containing sites 4 and 5. Now, if ATPP exists, then there is exactly 2 distinct haplotypes restricted to that set. Otherwise, you get a violation of the 4-gamete test, which Dan mentioned this morning. So you observe all 4 gametes, 0, 0, 1, 1, 0, 1, 1, 0. So no DPP. So these 2 conditions uniquely determine the root sequence as such. So that's nice. We don't usually know the root sequence in ordinary phylogenetics, but in this case we do. So how do we partition the input data into 2 subsets? Well, suppose that there is a DPP solution. Then you can convince yourself then, that there exists a HPP, haploid perfect phylogeny solution, satisfying one of the following 2 conditions. If the root sequences are not equal, then there's an edge connecting the 2 root sequences such that you can partition your tree into 2 sets such that each individual has exactly 1 haplotype in one set and its other mate pair in the second part of the partition. So individual 1 has 1 haplotype here and 1 haplotype here. Individual 4 has 1 haplotype here and 1 there. So every individual has this property. So you can uniquely determine the partition of your input matrix. What happens if the 2 root sequences are identical? In that case, we don't have such a nice partition, unique partition. But we have a situation like this, and the partition of your input matrix into 2 sets is not unique. For example, here you can either partition it like that or like that. So in this simple example, you have two choices. But one can actually show that those 2 partitions actually lead to the exactly same minimal DPP solution. So if a DPP exists for a given input data, then there exists a unique minimal DPP and a unique root sequence. So how do you determine the partition? Well, it's really simple. Once you have a tree, you just construct a associated bipartite graph. So you assign a vertex to each subtree adjacent to the root, and you label that vertex by the individual index. So this subtree here has haplotypes from individual 1 to 5, and you label it by 1 to 5. And this subtree 2 has haplotypes from individual 1 to 5, and you again label it by 1 to 5, and so on and so forth. And then you join 2 vertices in this associated graph if their label sets have non-empty intersection. And if the ATPP exists, then this must be a bipartite graph. But the bipartite structure is not unique because there can be more than 1 connected component. In this case, there are 2 connected components, so the bipartition is not unique. Either you have that or that. But the theorem, or the proposition I showed before, guarantees that it doesn't matter which bipartition you use, you end up with exactly the same DPP solution. So here's the algorithm. You first find the root sequences, and then you find whether there is a partition of the input data into 2 that's consistent with the algorithm I described. And you just concatenate the two data sets, HX and HY, to concatenate and also the root sequence, and try to build a haploid perfect phylogeny, and that will correspond to the diploid perfect phylogeny. Very simple algorithm, and it runs in linear time. So how would you do that for the genotype case? So Dan, again, was the first one to pose this problem. That is, how do you phase the input genotype data on a perfect phylogeny? And he did it for the case when you don't impose this condition on pairing of haplotypes to form individuals. He did it for the case when you just consider that they come from some some population, and they're not necessarily from individuals in an asexual organisms. So in that case, then has a nearly linear time algorithm and subsequently other people tried to come up with simpler but slower solutions, and eventually in year 2005, Dan and his student found a linear time algorithm for this problem, and also this group at Florida. In Florida also came up with a linear time algorithm for the problem. But the problem at hand is to phase the data, the input data, on a DPP, not HPP. So can we do this? So how can we do it? Well, at first it seems like a very hard problem, but it actually turns out to be trivial. Solution to this problem is quite trivial, and I'll show you how you can do it. I break up the problem into two parts. So first, I will try to construct a phylogeny using genotype data as characters. So I'll work with 1, 0, and 2s. I'll try to construct a genealogy for that. And then I'll use the genealogy to find a phasing solution. So some observations. When using the genotype data, recall that there are 3 genetic types, 0, 1, and 2. Zeros and 1s denote homozygotes, 2 copies of 0s, 2 copies of 1s, and 2 denotes a heterozygote, 0, 1, or 1, 0. And 1 mutation per site condition implies that the genotypic allele 0 and 1 cannot both appear in any column. Okay, that's a simple statement. It follows from the 1 mutation per site condition. And furthermore, when a mutation event occurs, then either you have a mutation which is from 0 to 2 or 1 to 0, 1 to 2, but never from 2 to 0 or from 2 to 1. Why? Because if recall what you have, 2 is 1, 0 or 0, 1. If you have a mutation event, then you're mutating to something you have already observed.
Because it can't.
So this immediately from this you can actually find construct an algorithm, simple algorithm to determine the root sequence. It's also unique. So check whether both 0 and 1 occur at any column. If so, then there's no DPP solution. If a column contains only 2s, such as here, then you assign 2 to the root sequence. If any column contains a 0, such as here, then you assign 0 to the root. If any column contains 1, then you assign 1 to the root. And that is the unique root sequence. And then what you do is you use that as your root sequence and build a haploid perfect phylogeny using 0s, 1s, and 2s as their character. And in each column, there are only 2 distinct characters that will appear. So it's equivalent to building a haploid perfect phylogeny. And we can use Dan's algorithm to build such haploid perfect phylogeny. So there's a linear time algorithm for doing that. And once you have a tree with a unique root sequence, then you just propagate. So what you do is you first arbitrarily phase the root sequence. You just do it any sequence, any phasing will do. And then you propagate this phasing down the tree. Again, you have choices here. Whenever there's mutation, you can either mutate the first one or the second one. So you can actually enumerate exactly how many phasing solutions there are. And this DPP serves as a data structure which encodes all phasing solutions. So yeah, so we have a linear time algorithm for the problem. So to recap, if a DPP exists for a given data set, then the root is uniquely determined, and there is a unique minimal DPP. And we have found linear time algorithms for both cases, for the phased input data and unphased input data. And this DPP serves as a data structure that includes all phasing solutions. So that's very clean, but how useful is it? So I implemented this algorithm in a software called clone tree, and I tried to use this program to analyze some real data, but I couldn't get access to any data out there. People are not willing to share their data. So I mentioned this paper by Sean and Martens, and she never replied to my email. So I couldn't analyze their data. So what I decided to do was to generate some simulated data. As Joe mentioned, it's just simple Wright-Fisher model and just forward simulation going forward in time. And I tried to generate some data violating this assumption of DPP evolution. So I introduced some amount of sexual reproduction and some amount of gene conversion, and try to see how often there is a DPP solution when there shouldn't be. So, I mean, the evolution is not compatible with infinite-sized model of mutation, but still, how much power do we have in detecting violation of such assumptions? So the first kind of simulation I did is the following. So I have some amount of sexual phase. So PS denotes the probability of having a sexual reproduction for each individual in the Wright-Fisher population. So I have a sexual phase for TS generations. And then I will turn off sexual mating and have certain time of asexual phase. So TA generations. And see whether any signature of past sexual reproduction remains at this time. Right, so this is what I said before. So I have a constant population size, non-overlapping generations. PS is the probability of sexual mating, and U is the probability of mutation. And TS is the sexual phase duration, and TA is the asexual phase duration. And here are the results. So these numbers are the proportions of data sets not admitting DPP solutions. So in some sense, they capture the signature of past sexual reproduction. And as you can see, if you have very small probability of having sexual reproduction, then you almost always find DPP solution, because there's no sexual reproduction, and hence it's compatible with DPP, and you should find DPP. So these are the percentages of violation of DPP assumption. And as you crank up the parameter PS, the probability of sexual reproduction, indeed, in this case, 100% of the time, you don't detect any DPP solution. So there's some signature of sexual reproduction we can detect. But the key observation here from this simulation study is that infrequent sexual reproduction in the population may be difficult to detect. Here, if you have infrequent sexual reproduction, then we just don't have any power to detect past sexual reproduction. So I think we need to be careful when making claims about ancient asexuality. Are they really asexual? Well, I don't know. I just want to point out that here I assumed a very small region in the genome. It's a single gene. As done in the past experimental studies, I considered only a single locus, single gene, small region, and if you use only a single gene, then you don't have much power. But I think we will gain more power as we get genomic data and the number of sequences increases. As you can see here, as the number of samples increases, then your power increases too. And another surprising finding is that the signature of past sexual reproduction decays rather fast with the time. So this is the time when I turned off the sexual phase and went into asexual phase. And after like 500 generations, the signature of sexual reproduction just decays. So how can we make claims about an organism being asexual for 100 million years or 40 million years? So we need to revisit this problem. Another type of simulation I did is mitotic gene conversion. So Dan talked about gene conversion this morning, but that was in the context of meiotic gene conversion events. But mitotic gene conversion events can also happen. So during mitosis, during self-cloning, you may have gene conversion events of this sort. And it is believed that these type of events are very important. It could be that the Ostracot's data, Sean and Martin's data, didn't show any signal in the overrepresentation of sequence differences due to gene conversion events. So how can we make statements, more precise statements about that? Well, we first need to develop some methodology that will incorporate asexual mating and gene conversion. Right now, there's no such methodology. In my work, I didn't include gene conversion or any kind of recombination. So I think it's timely that someone thinks about this kind of problems. So the upshot of this simulation study is that the probability of the existence of DPP solution decreases with increasing mean conversion track length. So this is a conversion track length. I remind you that corresponds to the length of the insertion here. So that's the track length. And as the track length increases, not so surprisingly, the power to detect gene conversion increases. And a second thing that's worthwhile mentioning is that even a moderate amount of gene conversion, by moderate I mean something that's comparable to mutation rate. This is the mutation rate, and this is the gene conversion rate. So if they are comparable, then it frequently leads to samples admitting no DPP. So one can actually detect gene conversion much more easily than sexual reproduction. So I think we have taken only a modest step toward studying asexual evolution for diploid organisms. And I think this is an interesting problem, and it also has important practical applications. So these are the questions that I posed in the beginning of the talk. So I will just show you here again to remind you. So I think these are really important problems. And molecular phylogenetic analysis should be able to provide some hints to answering these types of questions. Okay, right. So I think well, I'm supposed to go up to 4:20, but I'll finish up. Okay. So I've been trying to convince John Holzenbeck to join Power and try to look at this problem together, because he's the expert in statistical phylogenetics. What I have done here is just a combinatorial first look into the problem, but I hardly think that it's going to be the method to use. So we really need to think about more statistically sound methods for studying this problem. So these are some problems that one can work on. I think multi-state diploid perfect phylogeny is natural generalization. I don't think it's too hard. Polyploidy imperfect phylogeny, where you have multiple mutations, recurrent mutations, meiotic and mitotic recombination, gene duplication, as Joe talked about, some kind of coalescent process going backwards in time. And quite interestingly, I think this paper here illustrates that we really need to think about some models of natural selection that are applicable to asexual diploids. So this paper found something really astonishing. So it came out about a month ago. They found that a single gene, this diploid lotifers have two copies. One gene has adopted a particular function, and another copy has adopted a completely different function, and they work together to protect diploid lotifers from dying out during time of dehydration. So if there's no water, they will become desiccated, but it's not dead. They will live until you reintroduce water, and then it will be perfectly fine again. So it has somehow found a new way of adapting to these harsh conditions, although they are asexual. So what's the right model of natural selection for such individuals? I don't think there exists one yet, so we should think about that. Well, thank you. I just want to acknowledge Dan Gosfield for making very useful comments on this work, and thank you for your patience. I'm not so sure. I mean, these asexuals probably arose from joining of two haploid, which were asexual, and they joined. And I think the belief is that they became sexual and then lost sexuality. I think you can actually infer that from looking at its sister classes, which are asexual. Right.
Asexual development or lifespan. So when you look at several species, what kind of information do you think you will gain? Why you will gain anything at all?
Well, although I here didn't really specify the setting to which I should apply this method, but Meselson et al. They looked at different species. They didn't restrict themselves to a single species sample. They actually took something like 2 samples per species, and they looked at 4 different species. But still, even if you take samples from different species, you still need to deal with the evolution of asexual diploids, and the same kind of modeling should apply. I mean, I'm not, well, in the case you should also try to model speciation events, but I think the same idea of coupling the 2 genealogies should play a role there. Exactly how that's an open question. That's something that we should work on.